Assalamu alaikum. Today we want to talk about chapter 18, amino acids and proteins. Now this is part 2 of our course. We want to talk biochemistry. So we will cover the following topics here, as you can see, from 1 to 7 in part 1 and from 8 to 12 in part 2. So let's start. By definition of biochemistry is the study of molecules and their reactions in living organisms. Now the goal of biochemistry is to understand the structures of biomolecules and the relationship between their structures and functions. The pharmaceutical industry designs molecules that mimic or change the action of biomolecules. Also, nutritionists evaluate dietary needs based on biochemistry. Now we want to start with the first biomolecules called proteins. Proteins are polymers of amino acids. As you can see, there are 20 naturally occurring amino acids. Every amino acid contains an amino group and a carboxyl group and an R group called a side chain bonded to a central carbon atom, as you can see here. This carbon is called alpha carbon, so we have here alpha carboxyl group, and this is alpha amino group, and this is R called the side chain R groups, different for each amino acid, so that we can say that R identifies your amino acid. So always the central carbon is designated alpha. Each alpha amino acid has a different R group. So as I said, R identifies your amino acid. R groups may be hydrocarbons or may contain a functional group, as we will see in different amino acids. Now, two or more amino acids can link together by forming amide bonds, which we took in organic part, which are known as peptide bonds when they occur in proteins. You see, here we have an amino acid called valine and amino acid called cysteine. The alpha amino group with this reacts with the alpha carboxyl of this. We eliminate one water molecule to form this dipeptide, and this is the so-called the peptide bond. So dipeptide contains one peptide bond. Tripeptide contains two peptide bonds, and so on. Two amino acids form a dipeptide, three amino acids form a tripeptide, many amino acids together form a polypeptide. Now enzymes can be classification of proteins by function. Now proteins, they have various functions. If the proteins are enzymes, they catalyze biochemical reaction. If they are hormones, regulate body functions. Storage proteins like myoglobin. Transport proteins like serum albumin, structural proteins like collagen, protective proteins like immunoglobulin, and contractile protein like myosin and actin. So all these are functions of proteins. So proteins, they have various functions, as you can see. All the proteins in living organisms are built from 20 amino acids. Now, the amino acid proline is a secondary amine whose nitrogen and carbon atoms are joined in a 5-membered ring. So we start with the non-polar side chain amino acids. So again, here we have non-polar neutral side chains like alanine, the R group is CH3, glycine, the R group is H, isoleucine, this is the R group, leucine, this is the R group, methionine, the R group contains sulfur, phenylalanine, the R group contains aromatic ring. This is proline, is very specific. The R group is cyclic, as you can see, and this is known as secondary amine. The tryptophan, the R group is called an indoor ring and it's aromatic. Valine, the R group is isopropyl. So here we have 9 non-polar amino acids. Now The second class called polar neutral side chain. Polar neutral side chains like asparagine. Here they are group CH2 with this amide group. Cysteine CH2 attached to a thiol group. This SH called thiol group. Glutamine similar to asparagine but with an extra CH2 here. Serine similar to cysteine. Instead of sulfur we have oxygen. This is serine. And then we have threonine, also contain OH group. And here tyrosine aromatic ring with OH group on it. These are 6 polar but neutral side chain amino acids. So by now we have 15 amino acids. The remaining 5 amino acids are classified into acidic side chains and basic side chains. The acidic side chains are aspartic acid and glutamic. So they have similar R group, but glutamic has an extra CH2. The basic side chains like arginine with this guanidinium group. Lysine contains this non-alpha amino group if this carbon is alpha, beta, gamma, delta, epsilon. So this is known as an epsilon amino group. Whereas histidine here, the side chain is a 5-member ring called imidazole. Now, why they call these acidic side chains? Because they have negatively charged R group at physiologic pH, and these are termed basic because they have positively charged R group at physiologic pH. Now, the 20 amino acids that make up proteins are classified as neutral, acidic, or basic depending on the nature of the side chain. As we saw, the 15 neutral amino acids are further divided into those with non-polar side chains and those with polar functional groups, such as amide or hydroxyl groups in their side chains. The sequence of amino acids in a protein and the chemical nature of their side chains enables proteins to perform their functions. Now, we have type of forces that exist in proteins. Non-covalent forces act between different molecules or between different parts of the same large molecule. The non-polar side chains are described as hydrophobic, mean water hating or fearing, and to avoid aqueous fluids, non-polar side chains gather into clusters to create a water-free environment. So, for example, we find hydrophobic amino acids in the core of a globular protein, whereas the polar acidic basic chains are hydrophilic, water-loving. They are found on the surface of globular proteins exposed to water environment. Here, attractions between water molecules and hydrophilic groups on the surface of folded proteins impart water solubility to the proteins. Because globular proteins are soluble in water, due to this fact, all the hydrophilic groups are on the surface of the globular protein and they interact with water. Now, amino acids contain both an acidic group and a basic group. Now we want to talk about acid-base properties of amino acids. Now, these two groups can undergo an intramolecular acid-base reaction to form a Zwitten or a neutral ion with one positive charge and one negative charge and is thus electrically neutral. So whenever, for example, you dissolve an amino acid in water, it takes this ionized form. This is the ionized form. This is called the Zwitten. Zwitten is the form of amino acid in which the net charge on the amino acid is 0. Plus 1 minus 1 is equal 0. And this is known as Zwitten ion. In acidic solution, amino acid Zwittens accept protons on their basic carboxylate groups to leave only the positively charged amino groups. Now, in basic solution, amino acids veterans lose protons from their acidicness groups to leave only the negatively charged carboxylate end. So, if we have, for example, this general form of an amino acid, here you see this form predominates in acidic solution. It is fully protonated, as you can see, at low pH, and the net charge is +1, and this is called cationic form. Then this proton can be lost here to form the neutral or the isoelectric Zwittern with zero net charge. Then it can lose this proton from the alpha amino to get the anionic form with a -1 net charge. So simply, amino acids can be titrated. They can act as acid or base because they contain these groups. The isoelectric point Pi describes the pH at which a sample of an amino acid has equal numbers of positive and negative charges. At this point, the net charge of all the molecules of that amino acid in a pure sample is 0. The Pi for each amino acid is different due to the influence of the side chain. Now, a few amino acids have isoelectric points that are not near neutrality, or pH equal to. Now, how to determine this Pi? You add pKa1 plus pKa2 and divide by 2 to get your Pi value. Now, side chain interactions are important in stabilizing protein structures, thus it's important to be aware of their charges at physiological pH. Isoelectric points influence protein solubility and determine which amino acids in an enzyme participate directly in enzymatic reactions. Acidic and basic side chains are particularly important because at physiological pH, these groups are fully charged and can participate not only in ionic bonds within a protein chain, but also transfer from one molecule to another during reactions. Now, handedness. Now, the mirror images of your hand cannot be superimposed on each other, as you can see here. One does not completely fit on the top of the other. Objects that have handedness in this manner are said to be chiral, meaning hand. Now, alanine amino acid is a chiral molecule. Its mirror images cannot be superimposed. As a result, alanine exists in two forms that are mirror images of each other: a right-handed form known as D-alanine and a left-handed form known as L-alanine. And as you can see here, they are mirror images of each other: left-handed L-alanine and the D-handed L-alanine. So we can say alanine is chiral molecule. Chiral, not achiral. Chiral molecule because this carbon is surrounded by 4 different groups. So it is chiral. So you omit this E. Alanine is chiral molecule because of this. However, if you take propane, an achiral molecule, you see, because this carbon is not surrounded by 4 different groups, these two groups are similar and these two groups are similar. Now, carbon forms 4 bonds oriented to the 4 corners. In alanine, the central carbon atom is connected to 4 different groups: carboxylate group, an H atom, alpha amino group, and a CH3 group. Such a carbon is referred to as chiral carbon atom or chiral center. Then we have, as you can see, 4 different groups. 4 different groups here. So alanine is chiral. However, propane does not have 4 identical groups, so it is achiral. Then distinguish between chiral and achiral. The 2 mirror image forms of chiral molecule like alanine are called enantiomers or optical isomers. Optical because of their effect on polarized light. Enantiomers are one kind of stereoisomers, compounds that have the same formula and atoms with the same connection but different spatial arrangement. Enantiomers are non-superimposable mirror image isomers. As we saw, alanine, D-alanine, and L-alanine, they are enantiomers. Pairs of enantiomers have many of the same physical properties, the same melting point, solubility in water, isoelectric point, and density. Now, pairs of enantiomers often differ in their biological activity, odors, tastes, or activity as drugs. 19 of the 20 common amino acids are chiral. Only the L enantiomers are found in proteins. Even 19 of the 20 common amino acids are chiral. The only achiral amino acid is glycine. Why glycine? Because glycine, the R group is a hydrogen atom, which is repeated. And in nature, the L enantiomers are found in proteins. And you cannot find the D. The D enantiomers are found مثلا in bacterial cell wall, in the structure of some antibiotics, etc. Now, protein primary structure. The primary structure of a protein is the sequence in which its amino acids are lined up and connected by peptide bonds. You see here, as I showed you before, this is from R1 to R4, this 4 amino acid residue. So this is a butapeptide with 1, 2, and 3 peptide bonds. And here we have on the left, this is called the N terminal end, and this is called the C terminal end here. This is protein backbone, and this is the side chains. Now, the amino acid side chains are substituents along the backbone where they are bonded to the carbon atoms, as you can see here. Even R are always bonded to the alpha carbon atom. Now, the carbon and nitrogen atoms along the backbone lie in a zigzag arrangement with tetrahedral bonding around the alpha carbon atoms. Now, the carbonyl group double bond electrons are shared with the adjacent CN, as you can see here. This sharing makes the CN bond sufficiently like a double bond that there is no rotation around it. So this is the peptide bond. This is not a pure single bond, because of this lone pair can move here and this double bond can move here to give you positive charge on nitrogen and negative on oxygen. And this is called resonance structures. Now, because of this double bond character, there is no free rotation around the peptide bond. Peptide bond is planar and rigid due to this resonance stabilization. The carbonyl group, the NH group bonded to it, and the two adjacent alpha carbons form a rigid planar unit, as you can see here. This is the amide plane, this is peptide bond, and peptide group. Now, again, peptides and proteins are always written with the N-terminal amino acid, also called N-terminal on the left, as I showed you here, left, and the carboxyl terminal amino acid C-terminal on the right. The individual amino acids joined in the chain are referred to as residue. Here we have R1, R2, R3, R4. These are called amino acid residue. A peptide is named by citing the amino acid residue in order, starting at the N terminus and ending with the C terminal. So we can draw an arrow like this with its head here. So we call this the direction of the chain, from N terminal to C terminal end. The primary structure of protein is the result of amino acids being lined in precisely the correct order. So crucial is primary structure to function that the change of only one amino acid can drastically change a protein's biological property, as in sickle cell anemia, which is the result of only one amino acid substitution in the beta chain of hemoglobin. We substitute glutamic 6 with valine at the same position in the hemoglobin molecule, especially in the beta chain, and this will cause sickling of red blood cells and sickle anemia results, because we replace the glutamate, a polar amino acid on the surface, with valine, which is non-polar on the surface, and this will cause sickling of red blood cells. Thank you. End of part 1.